Chapter 3, Kuby, 7th edition. This is going to be talking about receptors and signaling, both B and T cell receptors. And there's a lot of information in the beginning of this chapter on signal pathways. Signal pathways are covered a lot in cell biology. Hopefully you've had that a little bit. I'm going to recommend that you just breeze through and read a little bit about the first several pages where it's talking about signal transduction pathways. I'm not going to make you memorize each one of the different ones, but understand that there's some chemical or signal that's going to be amplified to get an effect out of the cells. So if you have a receptor or a ligand bind to a receptor on the surface of a cell, then that can stimulate all sorts of kinases, calcium releases, all sorts of cascades which turn on genes inside the cells and cause the cells to become more active or to reproduce or to do whatever the effector function is. I don't think you need to memorize those, so just read through some terms that jump out at me that you should know the name of. I'm not going to talk about Michaelis-Menten equations too much. Just understand that the ratio of ligands to signal is going to determine how fast the reaction can occur. So the stronger the signal, maybe the stronger the signal goes into the cell, and that can cause the cell to react one way. Where if you have a weaker signal, maybe it's going to be a little different. The term avidity is used to describe the strength of the collective binding interactions. So if you imagine a group of cells, the ones that are nearest the source of the signal are going to get a stronger response, and they may become plasma cells, where the B cells that are further back in the crowd are going to get a weaker signal, and that may cause them to go ahead and differentiate and become memory cells. So just look at it from the standpoint of what are the big picture items on some of these signaling pathways. You can look, ITAMs is a very common thing, so learn what an ITAM is. Kind of learn what a lipid raft is. You can Google it, but there's also some very good diagrams in here that will help you out. You're not going to have to memorize what each of these adapter proteins does. I'm sorry, unless you're working in this area of research, you would not need to know that. The PIPs, whether they're phosphorylated or dephosphorylated, the PIP is, it's phosphatidylinositol bisphosphate. You don't really need to know that for this class. Read through it, look at the overall signaling pathway, but I'm not going to ask specific questions about too much except what I've already pointed out. This chapter has a lot of stuff on the signaling pathways, and the main thing is something grabs a receptor on the surface of the cell, it sends a signal, the signal causes a reaction out of the cell. End of story. Pretty much, that's what it's about. We will talk a lot of detail about the structure of antibodies, how antibodies are made up, and what they do, what some of the effector functions of the antibodies are. But that'll be in another audio clip.